Welcome to the third and final installment of Chapter 3 of Anatomy Physiology 1. We're going to go a little bit more detail into the different kinds of organelles and structures that make up the inside of a cell. I mentioned in our last episode that it'll really help you know and understand the organelles if you compare what they do with what you do with your whole body, with all of your tissues and organs, because they're just one cell, and so they have just the organelles to do all the things that we do. So we have muscles and we have the skeletal system that supports our body and allows us to move around. The cell is just a sphere of phospholipid, so it's basically just a wad of fat. So you can roll it around, you can push it with protein fibers inside. You can change the outside by pushing protein fibers up and increase your surface area. You can disassemble and reassemble tubules made up of little subunits, and that's another way that you can move around. Just push it forward, disassemble the back end, bring it forward, and reassemble it, and start pushing things about. Of course, we talked about the proteins that hold the cells together. So we have hemidesmosomes and desmosomes. One of the cool things that you can do with a microscope is you can have a specific dye for a specific organelle. And in this one, you can actually see that they have a kind of a reddish dye for the nuclear material right there. And then you have some of the microtubules that are going to be staining yellow and others are staining white. So that's kind of neat to do fluorescent staining like that. And everything else is invisible. I've pointed this out before, but I want to point it out one more time because one of my students, one of these days, is going to solve this mystery. So here are your centrioles, and it appears that they are directing these fibers on how to assemble these microtubules. But plants do the same thing, and they don't have centrioles. 3 types of proteins make up the cytoskeleton. You have microfilaments, intermediate filaments, and the microtubules. So you're going to have the really smaller proteins, intermediate size, and the microtubules are so big that they're actually hollow tubes. Here's a summary table of the 3 different things and what they do. Microfilaments form a web throughout the cell, and it's made of mostly actin. When we do proteins, we're going to talk about actin and myosin and how they interdigitate and cause the muscles to contract and expand. So we'll be revisiting actin because it's very important. The intermediate filaments, you can find a lot of these within your skin cells. They're made up of keratin. So you're going to find this is also your hair, your fingernails, if you had hooves. And then last but not least, the microtubules, the ones we saw being assembled and disassembled. That area where we found the centrioles is called a centrosome. And plants have centrosomes without centrioles. So what do you do with microtubules? You use them as a pathway. And I have a video, hopefully you watched, that shows you motor proteins that actually appear to be walking. And they're carrying things up and down using the microtubules as like a highway or a tightrope walker. The microtubules also make up the center of the cilia and the flagella, allowing them to beat or wave, depending whichever you're looking at, and it makes the mitotic spindle. So when we talk about when cells divide, these little microtubules are going to attach to the chromosomes, and they're going to pull them to either end of the cell. So that's how the cells divide. They do it by using microtubules to guide the chromosomes to the correct end of the cell. So you don't end up with two copies of one chromosome on one side and missing on the other side. Here's that link in case you missed it. It's called The Inner Life of the Cell. And here is a motor protein pulling this along. Another amazing fact is that not only is your cell membrane fat, it's just phospholipid bilayer, to double the phospholipid bilayer, so is your nuclear envelope, so is the mitochondria, the lysosomes, peroxisomes. That's a little bag of peroxide, like what you put on a cut. Endoplasmic reticulum is phospholipid bilayers. Golgi apparatus is also phospholipid bilayer. So there's only a couple of things besides the cytoskeleton inside of your cell that is not phospholipid. So that's kind of amazing that where cells are fat and the stuff inside the cells are fat, and yet they do all the things that we do as a human being. So the ribosomes are not made of fat, nor are the centrosomes or the centrioles. And the basal bodies are just the base of a moving flagella or cilia. The nucleus is the easiest to see. It's the largest of the organelles inside of your body. And I guess if you wanted to compare it with part of your body, it would be the brain. It's the thing where you can read the genes inside and make decisions. You can make proteins. You can change things about the cell. So it's kind of the working center of the cell that decides what kind of cell is it going to be, and what will it do, how long will it live, and what will be its functions while it's alive. Now, it's important to know that all cells have the genes, the information to be any other cell. So the muscle cell knows how to be a bone cell. A bone cell knows how to be a cell that makes up the back of your eye. So all the cells in your body have all the genes, but once the cell has decided to be a muscle, then it turns off the part that tells it how to be a bone or a fat cell or whatever else. So it's just going to have on the genes that it needs to be a bone. Some cells don't have a nucleus. So the one that springs to mind are red blood cells. They start out with a nucleus, but as they fill up with hemoglobin, they start throwing out their organelles through the cell membrane, and eventually they also throw the nucleus out. So then that's why they look like they're kind of caved in the middle, because the nucleus is missing. There shouldn't be more than one nucleus within a cell. The exception would be in your muscle cells, because you're literally, when they talk about, I'm going to exercise and I'm going to get ripped and I'm going to have such big muscles, you literally do rip. I mean, that's not just a term. And so the muscle cells can fuse together, and then you can have multiple nuclei. So as long as you're not trying to divide with multiple nuclei, then you're probably going to be okay. But other cells, if they try having more than one nucleus, it usually indicates that they're a cancer cell. Once upon a time, in between teaching or during teaching, I worked in a laboratory that was studying breast cancer. And it's odd that I was doing that, and then later on in life, my mother passed with breast cancer. But you can see these cells. Here's a cell, and it has more than one nucleus. There's the nucleus, and there's the nucleoli inside of it. Here's one. That's not a normally shaped cell. So first of all, you're looking at weird shaped cells, and then the second, look at the nucleus. You've got multiple nuclei in there. These are actual pictures through an electron microscope that is showing you the nucleus. Here you can easily see the nuclear pores, the holes that allow passage through the membrane. There are two layers of the phospholipid bilayer, and the pores kind of hold the two layers together. This is the scanning, so you can look at the outside, and this is where you've sliced through the cell, so you get a cross section. You can look inside. You can see the nucleolus right there, and the plural is nucleoli. There's your cell membrane, nuclear membrane rather, right there, and you can see the pores. So this is to allow things to escape and things to come inside, and the nucleolus is making ribosomes. So here's a summary slide saying those things. Other things you can do with the nuclear envelope is you can attach the DNA to it. So imagine, of course, people nowadays have dryers, and probably unless you went to your grandparents' house, you've never seen a clothesline. But you used to go out to a tree, or you'd put a pole in the ground, and you'd tie a sturdy string to it, and you would hang your clothes out on the line. So inside the nucleus, you can use the wall of the nuclear envelope to attach your DNA, so you can spread it out and read it as genes. Now we're going to say this again in another chapter, but I want to just say it one more time in this one, because one of the things that confuses students is what is the difference between chromatin and chromosome. So if the DNA inside the nucleus is spread out loosely, it's chromatin. And that's the only form in which you can actually read the DNA. You can actually look at the genes and make gene products, messenger RNA, and go on and make proteins. Once you have taken the chromatin and wound it up in preparation for the cell dividing, we call it chromosomes. And it's the only way you'll see DNA unless you have special electron microscopes. But your body has the ability to wind up your DNA tightly enough that you can actually see it with your naked eye under a light microscope. Another thing you're going to see on the outside of the nuclear envelope is rough endoplasmic reticulum. So there are some things that the nucleus needs, and how handy just to have the machinery that makes it sitting right out there on your nuclear envelope. So the ribosomes are making some sort of protein, putting in the lumen of the endoplasmic reticulum, and it can move in and out of the nucleus. One of the things that always tickles me or frustrates me, depending on my mood, is they're always saying things like, the liver is the largest organ in the body. And then somebody else will go, well, no, actually the skin is the largest organ in the body. So in another slide, it said the nucleus is the largest organelle inside of a cell. And now on this slide, it's going to tell you that the rough endoplasmic reticulum is the largest organelle in the cell. So it depends on if you want something all in one wad, like the liver, or all in one wad, like the nucleus, or if you're going to take something that's spread out over a large area, like your skin is spread out over a large area. So if you took your skin off and wadded it up in a ball, it would be bigger than your liver. Also, you have skin inside your body, lining your blood vessels, lining your organs. So they're cheating a little bit there. And endoplasmic reticulum, some cells are very busy making proteins, so they're going to have more endoplasmic reticulum than others. So again, it's often the largest organelle, but you would have to go and take it all and wad it up in a ball and then compare it to the size of the nucleus. So other things that the rough endoplasmic reticulum does is it makes the phospholipids and the proteins that make up all the other cell membranes. So it's making its own self, endoplasmic reticulum, but it's also making Golgi apparatus, nuclear envelope, cell membrane, all the phospholipids that you need in order to have a cell. It also synthesizes proteins, and it sends them through the lumen over to the Golgi apparatus to be packaged up so that you can secrete it. So it's kind of like your blood vessels. It's your transport system throughout the inside of a cell. The rough ER and the smooth ER are basically the same thing, but the rough ER has ribosomes attached and the smooth ER does not. So there's not a big distinction between the two. So what do you do with your smooth ER if you're not making proteins like the rough ER? Well, you are synthesizing steroids, so you're taking cholesterol and you're changing it into the various hormones that you need in your body: testosterone, estrogen, progesterone, hydrocortisone, aldosterone. And it also synthesizes some of your lipids besides the phospholipid bilayer. It also detoxifies alcohol and other drugs that you take. And I'm talking recreational drugs and then the drugs that your doctor prescribes for you. Another thing you can do with your smooth endoplasmic reticulum is you're going to store calcium. So calcium is very important, and places where you use calcium constantly would be in your muscles. So we're actually going to call endoplasmic reticulum in your muscles sarcoplasmic reticulum, which confuses students. I mean, it's still endoplasmic reticulum, but because it happens to be in the muscle and it happens to be full of calcium, we rename it sarcoplasmic reticulum. People who live out on farms are familiar with the term cistern. It's a holding tank for water. So they looked at the endoplasmic reticulum and they said, oh, look, those look like cisterns. So you'll find more cisterns in the smooth than you do in the rough. And of course, I have to throw in a few of the jokes that they have about smooth and rough endoplasmic reticulum. So here is rough endoplasmic reticulum bemoaning the fact that he can't get a date because smooth endoplasmic reticulum is so smooth. Another one I thought was absolutely hilarious is a dress that Beyonce had, and I don't know if she realized, but it looks a lot like rough endoplasmic reticulum. So, ribosomes are made by the nucleolus. The nucleolus is that wad of dark material inside the nucleus. They thought used to be, they just said, well, it's some sort of a staining artifact, and then they realized, oh, actually, no, it's RNA that's making the RNA you need to make ribosomes. And it takes it, sends it out, and assembles it into ribosomes. Here's a cartoon picture that I pulled off of the internet. And to me, there are two subunits that make up ribosomes, and to me, this kind of looks like a Mexican sombrero. So you have the dome of the hat, and then you have the bottom part of the brim. And this particular picture kind of truncated it so you don't see it quite as pronounced. But there's a groove, like in the sombrero, where the brim is turned up, and you're going to have messenger RNA sliding through the groove. And then up inside the part where the head would go, in a sombrero, that's where the transfer RNA and the amino acids are used to make proteins. So we'll go over protein synthesis in another chapter, but be aware that the ribosome is a 2 subunit piece. It's made of RNA, it uses RNA, and it makes proteins without the use of protein enzymes. So it was really an eye-opener to figure out how ribosomes worked. So how can I tell the difference between the Golgi apparatus and the endoplasmic reticulum? They both have cisterns, and they're both made of phospholipid bilayers. Well, if you look at the Golgi complex, it is a series of stacked closed-end bags, whereas the endoplasmic reticulum branches out and you don't see it closed off at one end or the other. So not only does the Golgi apparatus pinch off and make little vesicles, but it also does tasks of its own. It can synthesize carbohydrates and proteins. Proteins are not usually made in an active form, unless perhaps it is a structural protein. But the ones that are going to be enzymes, you don't want to make them active, because then they'll start acting immediately because they're fired up and ready to go. So you usually make enzymes in an inactive form, and then you just cut off a little piece of it to activate it, or maybe you add a phosphate to activate it, or take off a phosphate. So the Golgi apparatus helps put finishing touches on proteins. And when the rough ER is busy making the proteins, some of them are going to go throughout the cell, but a lot of them end up going through the endoplasmic reticulum, ending up at the Golgi apparatus where they're pinched off into little bags. It can add carbohydrates to the proteins. So we talk about proteins, carbohydrates, and lipids as if they are unique or distinct things, but you can have proteins with sugars attached, you can have lipids with sugars attached, so you can mix and match these various macromolecules. Some of the vesicles that the Golgi apparatus makes are lysosomes, full of digestive enzymes. One of the cool things you can do if you've got a bag of enzymes like the lysosome, and you have a bag that you just got through endocytosis or phagocytosis where you ate a bacteria. So you bring it inside, you've got it inside of part of the cell membrane, pinched off as a bag. You can just fuse the two because they're both made of fat, and you can just push them together, they'll fuse, and now the digestive enzymes are free to digest the bacteria, which is inside the phospholipid bilayer, without eating the cell itself. Because you don't want to digest the cell, you just wanted to digest the incoming bacteria. So you can use its amino acids and various chemicals, amino acids, things like that, and recycle them and use them within our cells. So in this way, the lysosome kind of acts like your digestive system. So the cell membrane ate the bacteria, it pinched it off into a sac. So the cell membrane acts as the mouth or the way of getting food into the organism. And then the endoplasmic reticulum can carry things if it's small enough, or in the case of a lysosome or a vesicle that contains something to be eaten, like the bacteria, then you can slide it along on those microtubules until they get together and they fuse. So the lysosome would act kind of like your stomach, because you have digestive enzymes in your stomach, and then in your small intestines and your pancreas is where you have the rest of your digestive enzymes. So here is a real electron micrograph picture, and then this is the cartoon representation of it. So if you look, you have these closed end bags, and they appear in stacks like this, and you can see the vesicles blebbing off, pinching off. If a cell becomes overwhelmed by invading viruses or invading bacteria, or it gets too old to perform its functions, or maybe you've been in a tanning bed and you've damaged the cells irreparably, you have the autolysis of the lysosome, where it will then just eat the cell and destroy it. So it has the ability not only to help you digest your foods, but if you're no longer needed or you're a danger to the organism, then the cell will commit suicide. And again, I like cartoons. If you understand why these are funny, then you actually understand how the cell is working. So in this particular case, this is a cell, a bacterial cell, and this is a virus attacking the cell. And here is denaturation. This particular cell got too hot, the proteins in it changed shape. You can get parasites inside of your cells. And last but not least, this one is apoptosis. This is the cell killing itself. This is a cell dying. And you can tell that it is doing it because it starts making these blebs. It starts making these projections sticking out. And once you see a cell start doing that, you know that it's about to blow. It's about to die. When we do the unit on blood, we'll talk about how malaria gets inside of the red blood cells and takes up residence and has babies. And another meme that I thought was kind of funny. Here is two cells shaming another cell. You have dishonored yourself and your organism. I do not deserve to live. Apoptosis. Cellular harikari. Harikiri. Excuse me, mispronounced it. If you've ever had a cut, sometimes instead of putting alcohol on the cut, you'll put hydrogen peroxide on it and it'll start bubbling and foaming. If you have an organelle full of hydrogen peroxide, we call those peroxisomes. Some means a body. So these are peroxide-containing bodies. And if you put something inside with the hydrogen peroxide, then it's going to oxidize it. It's going to put the oxygen on it, and it will destroy it. Or in the case of alcohol, it will detoxify it. Because alcohol is actually poisonous to us. And people who overdrink alcohol can actually die from alcohol poisoning. I've actually lost some students that way. So don't drink to excess. And then, of course, we talked about other drugs that are being broken down. So if you add oxygen to them, it will change whether they work, maybe make them more potent, make them less potent. So you're going to find peroxisomes in all of your cells, but you're really going to find them in the liver because the liver is one of its main jobs is to detoxify things, and in your kidneys. I always think it's funny when I look at a electron micrograph picture and they go, and these are the peroxisomes, and these are the lysosomes. It's like, how do you know? It's kind of like if you got a whole bunch of bags of garbage sitting out there on the curb, you don't know whether there's cans in there or your neighbor's one of those weirdo psychos that kills people and chops them up and puts them in garbage bags. I mean, you don't know what's inside of there. But they're always like, oh yes, this is a peroxisome right there, and that's the lysosome right there. It's like, it's a bag. So about the only way you could tell is take the bag out and chemically analyze the contents. But anyway, they all look like bags, like that. And then this, you can always tell by those inner folds that you've got hold of a mitochondria. So those are mitochondria. Those are not lysosomes or peroxisomes. Here's another mitochondria here and another mitochondria there. Hopefully you remember that mitochondria were once free-living bacteria, and they have those folds inside. They're called crista, so they name them crista. And one of the things that they do not show you in this particular picture is they were, at one time, free-living organisms, and they have their own DNA. They have their own chromosome. So they have a little circular chromosome, which they're not showing you in either the cartoon picture or the actual picture. This is where they've sliced it open and looked at it with a transmission electron microscope, or a TEM. You can see that there are ribosomes in there. And the reason that I pointed out that the mitochondria have their own DNA is because we have now found some diseases that people inherit, and it's not because it was inherited through the mother, but through the mother's mitochondria. So that's kind of weird. So we now have mitochondrial genetic defects. And the function of the mitochondria is to be an endosymbiote, to live in harmony with us, don't harm us, just sit there and make ATP for us, and we'll give you food, we'll give you shelter, we'll get rid of your waste for you. And so here's one of the memes that I enjoyed. It is, if you want power, you can use a battery. If you want real power, you can have nuclear reactors. But if you want a lot of power, you need a mitochondria. And then here's the guy representing a mitochondria. And he says, you guys get paid? I guess mitochondria are like those people that stand by the side of the road and say, we'll work for food. Because that's what they do. Have to throw in another mitochondria joke. So here he is being a couch potato. Says, when are you planning on finding a job? Don't give me that excuse you don't have any energy. You're a mitochondria for Nucleus' sake. I've been teaching over 40 years, and oddly enough, I was one of the first reviewers of Saladin's book, his first edition, many years ago. And here's something new. I did not know that the basal body at the bottom of the cilium flagella used to be a centriole. So we're finally figuring out what they're doing, but they still say it plays an important role in cell division. But plants do cell division and they don't have centrioles. Here's a picture of a centriole in cross section, and you can see that it's a series of 3 microtubules in a circular pattern. Here's the cartoon version of it. And for our last slide, it's talking about other things that you see inside of the cell besides the organelles, besides the vesicles. So you can have glycogen granules. If you have a lot of sugar, then your body has insulin and you have receptors, and they will cause you to pull the sugar in. If you can get a receptor, get the insulin, then you can bring the sugar in. You really have a lot of these in muscles because you need some quick energy. Our skin, our eyes have pigment. So you have, in the case of your skin, you have melanocytes, and they make melanin, and it works its way up into the squamous epithelium, the keratinized cells, and so you end up with darker skin if you have more of the melanocytes turned on. You can turn them on by going out into the sun and getting a tan. Other kinds of inclusions you may find in a cell will be little droplets of fat. You always need a little bit of fat on hand so that you can make phospholipid bilayers. Then you sometimes have foreign things that you find inside of the cell. A virus may have gotten invited in because there was a receptor on the outside that matched it. When we learn about the immune system, you're going to learn about how the AIDS virus found a receptor on the helper T cells. And because of that, because it invaded the helper T cells, it can knock out your entire immune system. So that's why AIDS is so scary. You don't die from AIDS, you die because your immune system is gone and you end up dying of cancer or pneumonia or something that you can't fight off. And in the case of COVID, we talked about the ACE2 receptor. We need the angiotensin converting enzyme receptors, but unfortunately, this particular strain of COVID, which is one of the coronaviruses, has mutated to the point where that's where it enters the cell. You also find dust particles. There's not a brain going on in the cell, and so sometimes they engulf things that aren't something that they particularly needed to eat. In your lungs, if you look at somebody who has worked in a coal mine, you're going to find particles of coal that's been incorporated into their lungs. People who work with sand inhale the sand and they get the particles in their lungs, and it's called silicosis. Here are the lungs from someone who died of black lung disease. Look how much coal there is incorporated in their lungs. This is the pink color you would expect in a normal lung. That's it for Chapter 3. Join us for Chapter 4 when we start talking about DNA.